Mm. Hello, everyone. I hope you can hear me. Great. So, good morning, good afternoon, good evening, everyone, depending on where you are joining us from. I very much believe that there are a lot of people uh, joining us from different countries, regions, and continents. And so, very much welcome, everyone. My name is Gode, and I am a member of the ECOP program mentoring and training tax team, now called the webinar team. So we run the monthly, or we host the monthly webinars. And in general, this is the ECOP program webinars. If you don't know about the ECOP program, it is simply the Early Career Ocean Professionals program. So it is for building capacity among early career ocean professionals. And if we say early career, if you are in your, 0 to 10 years of uh, professional career in the ocean space, including students, then you are considered an early career ocean professional. And then for this program is the UN Ocean Decade Endorsed Program for the youth in the ocean space. Currently, uh, we have ECOPs from 168 countries. We have 6 task teams in the ECOP program, and we have 60 regional and national nodes. If you are lucky, there is already uh, an existing national or regional node. So we have ECOP Africa, we have ECOP uh, India, ECOP Canada, ECOP. So we have the regional and the national nodes. But if you are even luckier, then we do not have one yet in your country, and you have the opportunity to spearhead or lead one and be a national coordinator. You can reach out to us after the webinar or any time at all, and would be very happy to assist you or to help you to develop the national node and be a coordinator. Thank you very much again. Today we have the webinar on microplastics, and we have two wonderful ladies joining us from the US. I would quickly give the opportunity to the first presenter, Lonnie. Welcome, and please take over. Uh, as Lonnie presents, I would like to remind us that feel free to introduce yourselves in the chat. Tell us your name, where you are from. If you have an interesting fact or organization, something to share with us, feel free to share it in the chat. Feel free to also post your questions and comments in the chat. Thank you.
Hi, everyone. My name is Lonnie Matthew, and there's a lot to talk about in terms of microplastics and their broader impact. But the goal for this webinar is to really look at their applied presence. And so we want to take a look at how microplastics are both a wastewater and transport issue, so that tracking their journey from manufacture to dispersal gives us relevant context. And so to start, we really want to look at where they go from wastewater treatment plants. And so I will talk a bit more, not just about this, but also how these processes relate in terms to human consumption. So there'll be a bit of overview in terms of microplastics, more information on wastewater treatment plants and their mechanisms, followed by the regional application of the Chesapeake Bay. And so the reason why we really want to focus on this is because it uses a case study that we can apply not just to this bay, but to other bays who that are also receiving this amount of microplastics from wastewater treatment plants on a daily basis. And so to supplement that, we want to use computer modeling systems as a way to forecast where these plastics go over the span of 60 days. And I'll go into more detail about the current research going on in regards to this. So microplastics are exactly what their name implies. They're microscopic pieces of plastic. And so they're plastic particles specifically less than 5 millimeters in size. So if you can imagine your standard pencil, it's the length of the tip of the eraser on that pencil. So they're broken down into two different categories, being primary and secondary. Primary microplastics, for example, would be previously manufactured plastic. So think, for example, microbeads in facial washes and each individual speck of glitter that's used in, say, for example, eyeshadow or lip gloss. Secondary microplastics are fragmented or degraded plastics that have corroded over time. So if you think a plastic water bottle that started as one uniform piece, once it's discarded, it eventually corrodes down to millions of microscopic pieces that are then considered microplastic. And so these are the types of plastics that are commonly found in aquatic environments, and those mostly include polystyrene, polypropylene, and polyethylene. So each of these synthetic fibers and others that haven't been named yet are a valuable resource because they can be manufactured into so many different things like cutlery, plastic forks, sterile medical devices, which is extremely important in terms of being sterile and not contaminating any equipment, bubble wrap, and things that we are just so used to in our everyday lives. So while the use of plastics are common from tire wear and household cleaning products, two of the most overwhelming sources come from cosmetics and clothing made from synthetic material. And so I wanted to really touch on where these sources come from and how they end up in our waterways. So, starting with synthetic textiles, it is a widespread use globally. And so, taking it back in terms of history, there have been many different types of fibers that have been used for textiles throughout human history. So, that includes silk, wool, and cotton. And it first started as early as the Stone Age. And so, the first manufacture of rayon yarn, which is a synthetic polymer, uh, took place in 1905, and its production skyrocketed by 1920 because of how versatile and how easy it was to produce. So this was the predecessor of nylon, which then brought us to polyester. So if we think in terms of timeline, it originally started with rayon, which then developed further into nylon, and now polyester has taken over the majority of textiles that we see every day. And so it was 52.66% of world textiles that were made of polyester from 2000 to 2008, if you can imagine that. So the evolution from initial synthetic polymers being used as textiles being rayon to now the majority of textiles being polyester, this has been something that has evolved over time. And so also during the Industrial Revolution and onwards, clothing items that were once time consuming to make became mass produced with newer technology. So with the production efficiency being increased, a lot of these textiles are commonly widespread because of how easy and accessible they are. Also, synthetic textiles are quicker and cheaper to manufacture, which explains also, again, their overwhelming presence in production. So another thing that we might forget is that we think of textiles in use in terms of clothing, but they're also used for things like carpet, upholstery, and bedding. And so as of 2010, clothing production has skyrocketed with the use of this material and are widely supplied internationally, as mentioned before. And so unfortunately, because we use these products and they go through wear and tear, we wash them, the fibers that have been used to construct them are commonly found in aquatic environments. And so we'll talk a little bit more how they get from manufacture to our aquatic ecosystems. And so, of course, we have cosmetics. So think, for example, hand soap that has some type of exfoliant that's made of plastic. Each of those microbeads that are used to exfoliate get washed down the drain. And of course, we don't think about it because they're so small. And also, we're used to things that exfoliate. People use glitter. I know myself, I use glitter in terms of like lip gloss and makeup. These things are all individual pieces of plastic. And so with this constant demand of usage, so body washes, body scrubs, hand soaps, lip gloss, eyeshadow, all of the above, there's a constant output. So they're constantly disposed of once they have been consumed, unfortunately. And specifically because of their size and because they have already been manufactured at such a small size, there's no way for them to be reused. And also it would be an issue of contamination in terms of how intimate these microplastics are to us in terms of our bodies. So now that we kind of got an idea of how these plastics are really entering our waterways, there's two main routes that lead to environmental ecosystems. So we have household water, which is what we were talking about before in terms of laundry and also cosmetics that make their way into treatment plants by way of being gray water, which is another term for household wastewater. And then there's also another source being stormwater or runoff water that occurs naturally after rainfall or after snow is melted over winter. And so taking a look back at the gray water from households wastewater, it deposits itself into a sanitary sewer, which collects and is then transported to a wastewater treatment plant followed by drain water. Those are normally directly input into waterways from storm drains. So there's a lot less discretion in terms of what types of things can flow away from the drain, the runoff water. So again, we want to think of how these treatment plants process this water that comes from households specifically. And so primary wastewater treatment plant methods includes the removal of large solids and grease. Secondary uses microorganisms to organically decompose matter and organic matter in that remains after primary treatment. And tertiary removes nutrients, pathogens, and fine particles, which includes microplastics. So as it goes further into treatment, the larger items get filtered out, followed by the ones that were able to bypass filtration, and ultimately the smallest parts or the more fine particles are able to be retained mostly. But there's a small amount of micron-sized microplastics that have been observed to still evade all of these filtration steps, despite all of these sophisticated methods that wastewater treatment plants use. And so in regional studies, it was found that nearly 90% of plastic particles are removed from effluents. So this is to account for the millions of microplastics that are treated daily. So if you can imagine the exfoliants in your hand wash that make it down the drain to a wastewater treatment plant, the vast majority of those plastics are still retained because of how advanced these filtration mechanisms are. However, as mentioned before, the margin for plastics to bypass are still present with millions of plastics being in effluent water post-treatment. So while there's the vast majority of plastics being retained from filtration, there's still that amount that, while small, is constantly flowing out from effluent water into aquatic ecosystems. And so microplastic fate after effluent dispersal is not widely known for the Chesapeake specifically. And this is a good case study because the Chesapeake Bay is the largest bay in North America, and we're able to see where these plastics go once they've been dispersed from treatment plants. And of course, after post-treatment discharge, they're reintroduced into aquatic environments. And so, as mentioned before, the dispersal of dissolved organic matter, bacteria, and the smallest of plastics still remain. So the plastics also undergo physical and chemical changes once they've been introduced to not only the water that the aquatic environment consists of, but also the effluent water, which carries, as mentioned before, bacteria and dissolved organic matter, which can then colonize these plastic pieces as well. And so when we take a regional look at the Chesapeake Bay, we can look here for Baltimore and Maryland, Washington, D.C., and then also Annapolis, which is 40 miles east of Washington, D.C. And finally, this is the southernmost part of the Chesapeake Bay that leads into Virginia. So these are the three highest concentrations of plastics, and they are positioned here with the highest population density being closer to Baltimore. Secondary or intermediate concentration levels were found south of Baltimore, which would explain the natural flow of the bay in this case. And then finally, the third out of the top three concentrated sampling sites was where the bay converges southward, where a lot of the water would pass through. So this paints a picture as to where these plastics end up. And we can also think in terms of population density. So regarding back to the amount of people that live near these areas, the amount of water that needs to be filtered every day is in relation to how many people live there. So, for example, in Baltimore, they have a higher filtration capacity at wastewater treatment plants because they need to serve so many houses that use water every day, versus a more rural part where there aren't as many people in the region. There's a smaller filtration capacity due to the lesser need for wastewater treatment for each individual household. And so a little bit more on this, once again, mentioning that this was a study that studied the surface waters of the Chesapeake Bay at these different locations. And so these are the types of chemical characteristics along with the physical characteristics in terms of shape. So the vast majority of plastics were polyethylene, polypropylene, and polystyrene. Anthropogenic other includes variations, so we can pretty much say that's the other type of plastic. What I also wanted to mention here was nylon, PET, which is polyethylene terephthalate, and other natural fibers were also found. And in regards to natural, that would be something like hemp or cotton. So when we look at the types of shapes that are able to make their way into the Chesapeake, a lot of them are fragments and followed by films and fibers. So what we want to take from this is that fragments are the ones that are broken off and are irregularly shaped, which we can infer are secondary plastics as they are not previously manufactured to take on that shape. So what we can take from this is that all of these types of plastic shapes and chemical types are occupying the Chesapeake Bay, but we also want to note that this is not strictly from wastewater treatment plant. It's also including drain water and other sources like just pollution that's alongside a type of a body of water that makes its way further into the Chesapeake Bay. And so the type of things that can happen once they're introduced into the water is that they leave environmental impacts mostly on filter feeding organisms. However, they compound in toxicity. So, for example, adherent surfaces being microplastics, they have PFAS, pathogenic bacteria, and metals that can all cling to their surface. Because they have a smooth surface and also they are adherent, as mentioned before, they are able to withtake or serve as a surface for all of these things to pretty much colonize onto them, thus making their way further into the Chesapeake more toxic. And so they're often ingested by filter feeding organisms such as oysters. And so specific to this region, we think about the eastern oyster, which has been observed to ingest microplastics specifically. And so I just want to take some time to point out this figure here by Tang et al. They were able to do a study by exposing oyster tissues to microplastic particles. And so what they were able to find is that the impact of exposure to microplastic particles was that it caused histological damage or tissue damage to oysters that were exposed. So these are their natural control. So this is what their cells normally look like. But post-exposure, you can see that they've become really inflamed. And in fact, some have ruptured as a result of exposure. So if we take a look down here also, this is a microplastic particle that has been colonized by by biofilms. So a lot of different microorganisms can colonize onto them. And while from the naked eye it seems insignificant or harmless, these type of pathogens or bacteria can colonize plastics and are then circulated throughout the aquatic ecosystem and introduced to parts of the ecosystem that they have not been introduced to before. And so how do we apply this to computer modeling systems? We seem to take something that's tangible and in person, something that you can see with your own two eyes, to something that's more of a forecast. So we can talk a little bit more about that by coming up with a plan on how to approach it. So again, we're taking the Chesapeake Bay, and what we want to do is take a particle tracking model, which simulates each of these individual microplastic pieces that are being dispersed from wastewater treatment plants daily. And so we use the particle tracking model alongside CHESROMS, which is a regionally specific ocean modeling system, to simulate the hydrodynamics of high tide, low tide, and the ebbs and flows that come with a water system. And so the variation of particle characteristics are going to be observed with this study due to its presence in the Chesapeake Bay. So now to visualize this, we want to take three different wastewater treatment plants specifically. And so we chose Patapsco Wastewater Treatment Plant in Baltimore, Blue Plains in Washington, D.C., and Cambridge Wastewater Treatment Plant in Cambridge, Maryland. And so, as mentioned before, we wanted to highlight the gradient of population capacity, filtration capacity from each of these locations, with Patapsco having the highest density in terms of population and Cambridge having the lowest population density. Blue Plains would be intermediate between both of those extremes. So we wanted to take microplastic fibers of ranging sizes, so that would be 10 to 500 micron. And then, of course, the plastic types chosen are commonly seen from wastewater treatment plants in the Chesapeake Bay. So to narrow it down some, because there's so many plastics, we wanted to really highlight three types that are commonly seen as we were able to look at before. So that's polyethylene, polypropylene, and polystyrene. And we have listed here their respective size in terms of how they are able to settle and sink over the span of 60 days. And so to further assess this and to approximate this in the best way that we can, we're using Fortran, which is a type of coding language that aligns these equations so that they accurately represent what we would imagine these plastic particles to sink and behave like, if you want to use the word behave, over time, because they all have different types of densities. And so we use two papers, one from Dittmar et al. And one from Bagheri and Bonadonna, to use their variables in terms of defining the types of equations we should use when coding in our model to represent sinking speed and settling velocity. So each of these aspects are mentioned. And so each polymer has a different type of density, thus it sinks at a different rate. And so with shape and size also included, plastic spheres, for example, do not move like a plastic fiber in water would, and vice versa. So we really want to get all of those details into the model so that it best represents what we would expect of these particles. And so in this case, we used Stokes' law and several other papers that model microplastics in water and compared those equations to our fibers and fragments to verify its accuracy in calculation. And so using this previous model simulation as a blueprint, if you want to call it that, the most relevant study to this region is Lopez et al. In terms of computer modeling microplastics. So they simulated spherical microplastic fibers and observed depth and buoyancy. And they also applied beaching, which is where a particle can touch a land boundary and not be resuspended into the water. And so they found that 92% of all particles are retained in the Chesapeake Bay, but we wanted to compare this with ours as it is regionally relevant and adding on to the information that we were able to deduct from already. And so to boil this down into a research objective, we wanted to apply a coupled hydrodynamic particle tracking model of the Chesapeake Bay to determine what factors, specifically size, shape, density, biofilm, beaching, and vertical turbulence, whether they are critical for simulation of the fate and transport of these particles. And so what we have now is a visual of the beaching factors, which mentioned before, where when particles touch the land boundary of some type of shoreline, this is something that we are able to really take a look into once we can see the visualizations of that. And so just following up with hypotheses, so we would also expect smaller, lower density plastic particles to travel further than those with higher densities and that are also bigger. Following that, we would think higher salinities at effluent sites will result in lower sinking speeds and hence further transport than at sites with lower salinities. And so this paints a picture of how these particles travel. So this is our control visualization. So we have each of those three origin points for the Patapsco wastewater treatment plant in orange, Blue Plains wastewater treatment plant in blue, and Cambridge wastewater treatment plant in green. So this is what we would expect a normal drop of water to travel over the span of 60 days. There are no individual ramifications. There are no individual parameterization of these particles, and so they naturally ebb and flow as they would in the natural environment. So these specifically are not microplastic particles. This is more so to simulate and visualize where a drop of water would go over time. And so over this time, we can see that the Patapsco wastewater treatment plant has the most presence in the main stem, which is this larger part of the Chesapeake Bay, followed by Cambridge wastewater treatment plant particles, and finally with Blue Plains wastewater treatment plant. So another thing that we want to mention too is the topography of this of the Chesapeake is also limiting some of the transport of some of the particles. So, for example, the Blue Plains wastewater treatment plant in DC is having more of a delayed response in terms to its dispersal into the main stem of the Chesapeake Bay because it has to navigate along a lot of curves and edges before it reaches that part. And so now when we take our particles and apply 5% beaching in terms of a 5% chance of them touching a land boundary and not being resuspended into the water, we can see a stark difference from our control group versus our beached group for 5%. And so as time continues, we can see that what once used to be covered with particles from all types of origin points with each of the three origin points, there are a lot more that got stuck on some type of land boundary and are no longer able to circulate. So with this, we're able to see that even at 5% applied beaching, the majority of these particles do not make their way far into the Chesapeake main stem due to the fact that they're stuck on some type of beach. And so what we can take from this is that when we're thinking of where these plastics go, we would normally assume that they would go into the water. But what this is showing us is that there are a considerable amount going on beaches and other shorelines instead of deeper into the water. So following this, we wanted to increase the parameterization of our beached particles. And so we went from 5%, which was already a stark difference from our control group, and applied 20% for this as well. And so over time, initially, it seems as though it has the same transport and trajectory, but very few plastic particles actually make it into the main stem. And so as they work their way around each of its respective tributaries, we can see that despite them all having the same characteristics in terms of start time and other parameters such as 20% beaching, they are still unable to successfully take a large presence in the main stem. And so what we can take from this is that the parameterization is a huge contributing factor, especially seen with beaching. So we can see how the lack of particles that were once able to get further into the Chesapeake Bay are then retained on beaches. So we can now think using this as a forecasting tool as to where these plastics end up specifically on shorelines and how to address that as a pollution issue as well. And so throughout each of the three model runs that we looked at today, Patapsco Wastewater Treatment Plant has the most spatial presence by day 60, and we could see that a lot of the particles that were in the main stem were primarily orange, followed by green, which was Cambridge Wastewater Treatment Plant. And so size, shape, density, and polymer type will further influence their trajectory, which is something that is still being worked on currently. But with these preliminary results, we can already see the vast difference in terms of where these particles go. And so to summarize on the research aspect, this is the first study of its kind in terms of wastewater treatment plants and microplastics in the Chesapeake Bay. It provides clarity on the most important aspects for computer modeling simulations for this specific study. And then it also extends on information from previous studies, as mentioned before, from Lopez et al. So this is more of a computer modeling, forecasting takeaway. And then also we can think about it in terms of our real lives. So, in terms of applying it to our everyday lives, there's an excess presence of synthetic material. If you do laundry, lint is full of microfibers that are synthetic. And so once we have them in the dryer, we can imagine the amount of plastic fibers that are being dispersed from washers. So the constant presence of these fibers and microplastics in gray water are constantly being dispersed into wastewater treatment plants. The smallest plastics bypass those filtration systems and are redispersed back into the environment. And so now that we know this, why really does this matter? Well, we can now observe patterns to where plastics accumulate. So now that we know, based on the imagery that we looked at, a lot of these plastics actually don't get too far into the Chesapeake, but they do get caught on some type of beach. And so again, this is something to simulate what we would see in real life. And while the perimeters can be adjusted, this is what also is representative of the Chesapeake Bay. And so this is, there are implicit mitigation strategies that can be taken from this. So we can observe the highest concentration in flow and try to reduce overall exposure from what has been modeled. So where the bay converges, there can be types of mitigation strategies that can be pulled out from studies like this. This also demonstrates impact of plastic presence in relation to filtration capacity and population density. So as mentioned before, where there's more people, there's more water to be filtered at wastewater treatment plants. Where there's less people, there's less water to be filtered. And so when we take this, we can visualize how often and how concentrated microplastics are dispersed. Because, as mentioned, there were only 3 wastewater treatment plants observed for the countless amount of wastewater treatment plants that exist within the bay itself. That doesn't include other types of bays that exist globally, but we can imagine with just these 3 of how many particles are able to disperse from one specific wastewater treatment plant in its own. And so this can lead to management, sampling, and monitoring decisions that can be deducted from this type of information. And with that, I would like to say thank you for listening.
Great. Thank you very much, Lonnie. That was an amazing presentation. I just got some back comments that say the speaker is really good, by the way. Extend my compliments. So that was an amazing presentation. And then, so I would like to just launch a poll quickly, just so we understand where, just a little bit about our uh, audience and listeners, so to know where they are joining us from, especially, and what they do. So, I mean, before we have the second speaker, please respond to the poll on your screen just in a minute. So, are you an ECOP? I already explained earlier, if you are in the first 10 years of your career, um, then yes. How do you interact with the ocean, or in which field are you basically right now? Are you a student? Are you working in government? Where are you joining us from? Please respond to the polls and then we can proceed. There are a number of responses. So kindly respond to the polls on your screen. Yeah, I see some Ghanaians and Nigerians here. So, but in the polls, we do not have. Okay, yeah, now we have someone from West Africa, most from North America, and then Europe, Asia. Great. Oh, okay. We have a number of people who work in the microplastics domain, so that is really good to see. So I'm really sure you will be, adding some knowledge. And there's a question on the model for you, Lonnie, but I'm sure you would be happy to answer this later.
Okay, I will be sure to
So
answer the question.
yeah, everyone, right after Robin's presentation, we can have a little, chit chat and conversation at the end for some 5 minutes or 10 minutes, just to connect and also to answer some questions that you might have. So great. Most people are ECOPs and it's good to see. All right, so if you have not responded to the post, please do so in the next 30 seconds. I will end the poll. Thank you. All right, then. All right, so we have most people are ECOPs, which is nice. We have a few people who are not ECOPs, really great to see. Also, most people in the academic space and they are students, so really good to see also because this is very much more, thesis uh, focused and oriented. So it's really good to see that most people are in the academic space and they are students. And then we have people joining from North America, West Africa, Europe, and Asia. Really great to also see, and that most of the people or a lot of people work in the microplastics uh, domain. So really good to see. Um, yeah, so in one word, what do you think about microplastics? Uh, there are a few words here, so problematic, pollutant, issue, barrier, substance, small particles, biodegradables, destructive, health, danger, cross, emerging, important, marine, brain. Very interesting words, I must say. All right, then. Thank you all again. We can quickly move to the second presenter. So, Robin, please, if you are ready, you can start.
Hi, everyone. Give me just 1 second to share my screen. Okay. So, hello, my name's Robin Dudrick. I'm a graduate student at the Batten School and Virginia Institute of Marine Science, and I'm really excited to talk to you all today about microplastics. Really grateful to Lonnie for setting the stage. So, she talked about how microplastics get into the environment and forecasting where they go. And my research kind of comes in where she left off with how do we actually find plastics in the environment and quantify what's there and sort of validate the kinds of models that Lonnie's working with. So, to give you a quick overview of what we're going to be talking about, I'm going to define plastics a bit more in depth. I'm going to talk about some of the environmental sampling and detection methods that we use in the field of microplastics research, and then I'll pivot into my study, which is looking at plastic pollution in coastal bays, and I'll give you some preliminary results and some conclusions, both from my research and from sort of the broader discussion. So, what are microplastics? Lonnie gave us a definition to start with, but I want to get a bit more in depth, make sure we're on the same page. And before we can really talk about microplastics, we need to understand plastics in general. So, plastics are synthetic or semi-synthetic polymers, and a polymer is just a repeating monomer. And let me turn on my pointer here. So plastics can be made from all different kinds of polymers. And if we look at this figure here on the left, we have some of the standard recycling codes and abbreviations that we use in the US for these plastics. But this rightmost column is the monomer. That's that backbone that's going to be a repeating unit to make up plastics. And when we refer to plastics, both scientifically and just colloquially, we're usually referring to them by their polymer. So PET, polyethylene terephthalate, or polypropylene, that's the polymer that we're talking about. However, polymers aren't the only part of a plastic. There's a ton of chemical additives that go into plastics to get the end user desired result. So plastics have different textures and colors and purposes, and the way we can go from a handful of polymer backbones to a million different uses is through the chemical additives. So this figure here on the right is showing you some of the categories that are commonly used for chemical additives. And there's over 10,000 types of chemicals. And these can be things like colorants, stabilizers, flame retardants. They all serve a purpose, but we don't necessarily know which types and how much of each of these additives are going into that final product that we interact with. And so when we think of plastics, they're this sort of diverse combination of a repeating polymer chain and all this other stuff. And for example, all the pictures here on the screen are PVC, polyvinyl chloride, but they all look different and behave differently. So we've got sort of standard piping, we have tubing, we have buoys and fishing lures. And the way that these examples of PVC are going to behave are going to depend on their chemical composition, like we just talked about, what kinds of additives are in there. It's going to depend on their physical traits. So are they rigid? Are they heavy? Are they flexible, buoyant? What color are they? All of those things can affect how they behave in the environment. And then their exposure to weathering is also going to be pretty important. So if we look at these two images of buoys, this orange one is pristine, and this one on the right that's white and yellow or white and blue is a weathered one that's been out in the environment. So they obviously look different from each other, they feel different, and they behave differently when we interact with them as well. And this is really important when we think about microplastics. So I'm not going to touch on the primary microplastics at all. I'm just going to focus on these secondary microplastics, which are primarily generated through environmental weathering of macroplastics. And then they're fragmented over time with UV exposure, with wave action and friction and abrasion to become these secondary microplastics that are smaller than 5 millimeters in size. Excuse me. And I have these images here to give you a sense of just how variable microplastics can be. Less than 5 millimeters maybe doesn't sound like a big range, but it actually is when we consider something like this top picture showing plastic fragments greater than 1 millimeter. If you look at that, you can right away tell that that's not a natural or an organic particle. The color and the shape of it, if you pick it up, the texture, it's pretty obvious to most of us. That's a plastic. But if we look at this bottom picture here, these are microplastic particles that came from these same fragments up above. They were just ground down to be this finer size class. And now it's a lot harder to tell what we're looking at. We can maybe see some colorful specks in there indicating that this isn't just sand, but the vast majority of it, it's brown, it's fine. If we were to pick it up, we wouldn't necessarily know that it's not sand. And so this is highlighting both the diversity of microplastics and some of the challenges that come up when we try to study them. Because as we get into the smaller size classes, which are more and more abundant in the environment, we have a tougher time determining where they're coming from and what they are. I also want to take a moment to introduce tire wear particles. These are generated throughout the lifetime of a tire. As you're driving, your tire is going to be shedding off these little specks. That's why you have to get new tires throughout, driving a car, having a bike. And these particles are very similar to the sort of traditional microplastics that we talked about earlier, in that they can be different sizes and shapes, and they also are made of a variety of ingredients. So just like polystyrene isn't just polystyrene, it has chemical additives. A tire is not just rubber, it has all of this other stuff in there. And just like the traditional microplastics, they're usually proprietary formulations. So we don't entirely know what's in there and what quantities, and that makes it challenging to research them in the lab or in the environment when we're not precisely sure what we're actually looking for. So if there's one thing that you take away from this talk, I would like for it to be that microplastics are complex and they're diverse and they're really complicated and can be challenging to study. So when someone is talking about microplastics, if you're reading about it, I'd really like for you to take a moment to think, well, what do they mean when they say microplastics? What polymer are they talking about? What additives? Do they care about the original source, where it came from and what it was used for? What size are they talking about? The morphology and color, all of these things are going to influence, where we find it, what effect it has, essentially why we care about it. So microplastics are really diverse and they're a category of pollutants. The word microplastic doesn't refer to just one thing. All right, so how can we study and sample such complex pollutants? How do we go about this? Well, there's a lot of different methods, as you might expect, and I'm going to walk you through some of them, just a brief overview. So first of all, when we're talking about plastics in the environment, we have to define what our environment is, what matrix do we care about? And I'll give some examples for some common methods for each of these environments here. So if you're talking about water samples, then maybe you're going to do a surface tow with a net. Maybe you're going to just take a grab sample, a scoop of water, or maybe you're going to pump up water from deeper within the water column. If you're interested in the sediment, you can take a sediment core to get layers and maybe look at what's going on over time or across different sediment compositions. Or you can take a grab sample. This is called a Van Veen grab, and it just scoops up a big chunk of sediment. I'm not going to talk about air because I don't actually research microplastics in the air, but plenty of people do, and there are methods for that, and you're welcome to look into it if you're interested. So the last one I'll hit on here are plants or organisms. Lots of people are curious about microplastics in the fish that they're eating, or maybe in various aquatic or even terrestrial plants. And usually we're going to sample these by just physically, manually collecting whatever species or organisms that you're interested in. And that's great. Now we have our samples, but we can't just throw that onto the instrumentation and analyze them. We have some processing steps to actually be able to look at the plastics in our sample. So I'm calling this concentration because regardless of where you're sampling from, you're usually going to have a bit of your sample that you are interested in, that you care about, in this case, microplastics, and a whole lot of your sample that you don't care about. That's the water or the sediment that is all mixed in with it. That's going to add noise to your final output and really mask the signal of the microplastics of interest. So we can go through these concentration steps to try and concentrate the microplastic we're interested in and remove that extraneous matrix that's just going to kind of get in our way. So three common methods of doing this are digestion, density separation, and filtration. These are some pictures from my workflow in the lab here that we'll just briefly go through. So up on the left, this is a density separation device, and you can put your sample and some dense fluid in the bottom chamber. We shake it up, we let it settle, and it's going to separate out the more buoyant particles on the top from the more dense particles on the bottom. And this is a really useful tool because we know the density of many plastic polymers of interest. Lonnie even showed you some in her slides. So we can create a solution of a specific density to target the polymers we're interested in, and that's going to help drop out a lot of the contamination from that matrix that we're not interested in and try and pull out just those plastics we care about. However, this is not a perfect process. So if we look at this filter here that came from that top chamber, you can see that there's some organic material, some plant debris, because those are also more buoyant than the solution that we put them in. So the next step then would be digestion. And digestion is just eating away that excess material that we're not interested in. There's lots of different reagents you can use. They can be acids, bases, enzymes. I use hydrogen peroxide. And the point is just to help clean up our sample a little bit more, get rid of all that noise that we don't need. After that, we can do our final filtration and get our final sample that we're going to analyze. So on the right here, I have two sediment filters on the top and bottom, and then a water filter in the middle. So you can see that our end result is going to be variable, and that's really common in microplastics research. But this is now clean enough for us to start to do some analysis, which is the fun part, identifying and quantifying the microplastics in our samples. There are a couple common techniques for doing this, namely microscopy, spectroscopy, and spectrometry. And if you're not a chemist, don't worry, we're going to walk through this really briefly here, talking about some of the common detection methods that are used in the field of microplastics research. So the first one, our simplest and most accessible method, is just visual analysis through microscopy. So you're manually picking particles that you can see under the microscope, and you're determining whether you think those are plastics or organic particles. And we can use a hot needle test to assist with this, where if you put something hot on a piece of plastic, we're probably all familiar that it's going to melt, but organic material doesn't behave that way. So that can help us kind of validate our visual identification. And at the bottom here, we can see some examples of different types of microplastics that have been visually identified from samples. And this can work really well with larger microplastics, and especially with things like these blue threads, these purple fragments that are just very clearly not organics. But it gets a little trickier when we get into fragments and nurdle-looking spheres that maybe it's a grain of sand, maybe it's a broken shell fragment. It becomes a little bit harder to be confident in our identification of these particles as synthetic rather than organic. So that leads me to chemical analysis. We can use more advanced tools to actually identify the polymers in our samples and confirm we're looking at what we think we're looking at. So one of the really common ways to do this is spectroscopy, such as Fourier transform infrared or FTIR, or Raman spectroscopy. And these are using different wavelengths of light to vibrate and stretch the molecular bonds within the sample and output an IR spectra. And these IR spectra are unique for the composition of these polymers. So on the right here, I'm showing you some reference spectra for some of the very common polymers that we see in the environment, polyethylene, polypropylene, etc. And we can see that they have these valleys that are unique to each polymer type. So by taking an IR spectra of our unknown sample and comparing it to a known reference library, we can then positively identify what polymers are present in our sample. And this is really useful. It's an improvement over microscopy because it gives us that confirmation, but it does have some downsides in that it doesn't work as well with weathered plastic since it is a sort of surface level analysis, and it's not 100% accurate. We can also use thermoanalytical spectrometry as a form of chemical analysis, such as pyrolysis, gas chromatography, mass spectrometry, or PI-GCMS. And this is using extreme heat to thermally decompose our samples. So we're pyrolyzing a liquid or a solid sample into its gas phase, which is then analyzed using gas chromatography mass spec. And that again gives us this kind of unique fingerprint by separating out molecules by size and their mass to charge ratio and giving us a pyrogram, which looks a little something like this. Now, the main thing to take away here is that the output, while different from an IR, works in a similar manner where we get peaks for our unique samples, and we also get peaks for reference standards. And we can compare those to each other to positively identify which polymers are in our sample. And a really cool advantage of using something like PyGCMS is that we're not only identifying the polymers in our sample, but we can quantify how much is in there as well. So if we briefly compare these different types of analysis, there's pros and cons to each one, and there's no one size fits all approach. They're going to vary in their cost. They're going to vary in what size fractions they're able to look at. Like I said, microscopy is better for larger things. Spectrometry can get into the smaller size classes that are more looking like sand. They also require different amounts of time for sample pre-processing or the analysis themselves, and they'll give you different outputs. Some of these will tell you about the size and the shape and the number of fragments, whereas others will give you the polymer ID or the quantity. So it's really going to be a trade-off here. And at the end of the day, how do we measure microplastics? Well, it depends on the resources available to you and the question of interest. So these two factors are really going to determine which method you're going to choose for the sampling and for the analysis that we just walked through. And one of the fun challenges of microplastics research is that you do have so many options out there. So it can be a little hard to compare results across studies if people didn't use the same methods as you, but we're aware of this as a research field, and we're trying to find ways to make results more comparable and to make microplastics research more accessible. All right, so with that, I'm going to pivot to talk about my study specifically. So as I said, I'm a master's student and I'm researching microplastic pollution in coastal bays. So once plastics get into the environment, where do they actually go? Where are they concentrating? And what types of plastics are there? Those are the kind of baseline questions that we need to answer before we can start to think about ecosystem impacts and what it means for the greater ecosystem as a whole. So my study was motivated by this idea that coastal ecosystems are important for the global plastic budget. We know that they receive input of pollution from both ocean and land-based sources. So they're this hotspot for plastic pollution. And research is showing both through modeling and from on-the-ground studies that plastics are indeed in every environment we look for them, including these coastal ecosystems. And microplastic research is increasing, and over the past few years, we've seen this great uptake of microplastics research in coastal ecosystems, which is wonderful. However, ecosystems aren't being equally represented across the literature. So if we look at this map here on the right, this is showing you seagrass ecosystems specifically across coastal habitats globally, and we're showing their abundance here. So if you look at the amount of this map that's covered in dark blue, we can see that seagrass ecosystems are highly abundant all over the world. Yet that's not really reflected in the literature, where mangroves seem to be the predominant coastal ecosystem that's been studied thus far. Quick sidebar, what even are seagrass meadows? Well, seagrasses are plants that grow underwater. So if you just think of grass that grows on land, basically the same thing, simplifying it, but basically the same thing underwater. And large areas that are covered by seagrass are called seagrass meadows, again, similar to just a meadow that's on land. And ecologists have been studying seagrass meadows for a long, long time. So we know that they're really beneficial coastal ecosystems. They provide a lot of services, both to the aquatic and marine environment and to humans. So seagrass meadows are big sources of primary productivity and carbon sequestration. They provide nursery habitat for species that are both economically and ecologically important. They reduce wave action, which can be impactful for reducing shoreline erosion. And they can trap organic particulate matter from the water column and sequester it in the sediment. And it's this final ability of seagrass meadows that makes them of particular interest when we're thinking about microplastic research and microplastic intrusion into coastal ecosystems. So both lab and field studies have shown that seagrass blades are capable of trapping microplastics, but the jury's still out on whether seagrass meadows are trapping more microplastics than nearby bare sites. We don't know if they're actually having an effect that's different from other locations or if this is just, a factor of, well, microplastics are everywhere. So that leads to my research question. I want to know if restored seagrass meadows can actually serve as a sink for microplastics compared to nearby bare sites. So my study takes place on the east coast of the United States, out on the eastern shore of Virginia. We have these wonderful barrier islands that create these coastal lagoons that make really lovely habitat for seagrass meadows. So I work down here on the southern tip of this peninsula in the South Bay Meadow. And for my methods, I took sediment and water grab samples from both meadow and bare sites across the growing season in summer of 2025. I then concentrated the plastics from my samples using those methods we talked about earlier, where I did density separation, digestion, and filtration. And then I analyzed my samples using PyGCMS, and I had 8 target polymers of interest. So these are polymers that are commonly produced and commonly found in environmental studies. And down here at the bottom, this SBR is our proxy for tire wear, which I mentioned is considered a type of microplastic, but is not always included in studies because it's a little challenging methodologically to incorporate. So we went ahead and threw that in there as well. Now I'm going to walk you through some preliminary results, and I'll caveat this by saying that this is about a quarter of my data. So I'm excited to show you what might be going on, but it's very much a might be going on situation. We have a lot more data to add here. So to get started, I just looked at the presence and absence of polymers, both by site type, meaning bare versus seagrass, and the environmental compartment, meaning sediment or water, just to see what's showing up. And there's 2 main takeaways from this figure. The first is that there are more observations of microplastic in sediment than water samples, as we can see by comparing these top 2 donuts to these bottom 2. And the other takeaway is that polymer composition is quite variable across both site types and compartments. So the amount of colors we see per donut and the thickness of those wedges is different across each one. We're seeing high variability, which is pretty common in microplastic studies. We can go a step farther than presence and absence, though, when we use PyGCMS and actually look at the polymer concentrations in our samples. So we're looking at the concentration in micrograms per kilogram for sediment samples and micrograms per liter for water samples. And each panel here is representing one of those polymers of interest that I showed earlier. So there's a couple takeaways from this as well. I know there's a bit going on here. So first, there are no observations of PMMA or SBR, which again is that tire wear proxy, in any of the samples that we've looked at so far. We're also seeing that polycarbonate, polyethylene, and polypropylene are the main contributors to polymer concentration, whereas these 3 polymers aren't really doing too much. And finally, if we look at the circles versus the triangles, we can see that there are more microplastics in the sediment than the water samples. All of our triangles are hanging out at the 0, whereas our circles for sediment are populating the figures. So if we throw that all together into one graph, now it's all on the same scale, try and pull out any patterns, we don't really see any patterns. There's high variability in concentrations across sites for all of the different polymers, and that's kind of to be expected. As I said, variability is really common in microplastic studies. And again, this is only a quarter of my data. So it'll be really fun to see if patterns do start to emerge as we flesh out this graph some more, or if there's just no trends to be seen. One of the joys of research is that we just don't know. So I'll leave you with a couple of conclusions, both for my study and more generally. For my research, the 3 main points that there's more microplastic in sediment than water, which is supporting the hypothesis that seagrass meadows are trapping microplastics in a similar manner to the organic particulates, and so they may actually be serving as a sink for them, which could be beneficial to the ecosystem if they are keeping microplastics out of the water column and sort of bearing them away. Or it could be detrimental to the ecosystem, since we know there are lots of benthic organisms that dwell in the sediment of seagrass meadows. And if we have higher concentrations of microplastic in the sediment, they're at a higher risk for exposure. And we just don't really know what interactions it may be having and what those impacts may be. So jury's still out on that one. Our second finding, the three main polymers that are contributing are polycarbonate, polypropylene, and polyethylene. And this is actually not too surprising. These are some of the most commonly produced and used single-use plastics, so we often see these in environmental studies. Finally, we did not detect SBR or that tire wear proxy in any of the samples so far. So this could either mean that it's truly not there, which would be great for the ecosystem, and wouldn't be too surprising given that this seagrass meadow is separated from land, which is of course the source of tire wear, by some marsh and some oyster reefs. It has quite a bit of distance to travel and habitat to get through before it would make it to the meadow. Or it could mean that there is tire there and we're just not catching it with our detection methods. So we'll see as we get the rest of the data in if it starts to pop up anywhere. Then for some broader takeaways just from this talk in general, not specifically my research, microplastics are diverse pollutants that are complicated and they can be measured in a lot of ways. So we have to think really critically about what we mean when we say microplastics and when we talk about microplastic research and impacts. And the accessibility to analytical techniques can be a barrier for people who are interested in doing these kinds of studies. So that's something that as a field we really need to work on. And then once microplastics get into the environment, their fate is hard to predict. Again, they're so complicated. There's so much going on. Lonnie did a great job of talking about this with her modeling. We don't exactly know where they're going. So that makes it challenging to guess what the ecosystem health impacts are going to be if we don't know where they are and we don't know what they are. So with that, I'd just like to thank my lab, my collaborators, and my funding sources, and I'd like to thank you all for attending and giving us some of your time. Stop sharing my screen.
Hello. Yeah, thank you very much, Robin. That was a very interesting presentation as well. And really, thank you. Lonnie, would you like to address the question that was posed to you earlier?
Yes. So, hey, Tejaswini, am I pronouncing your name right? Hopefully, I'm not butchering it. Um, I did want to say that Chessrooms does use, there are boundaries in the model. So it's an open ocean boundary near the Atlantic Ocean. So that's further south. The visualizations in the GIFs didn't really show that bottom area because it wouldn't fit horizontally. But the boundary for the Atlantic Ocean is also in the model along with river boundaries too. So between those two, it's trying to simulate those edges specifically for the rivers and tributaries that the particles are starting at. And then also in terms of the grid usage, I put it there in the chat. There's some rectilinear grids with uniform horizontal spacing. And so this is pretty much what chess has been composed of and how we're able to block off those boundaries.
Um, does that answer your question? Not sure if there's another question, but does anyone have any other question for either Lonnie or Robin? So, Robin, I want to ask, what is the full meaning of SBR?
Yes, good question. SBR is an abbreviation. It stands for styrene butadiene rubber. And so that's a polymer standard that we're able to get that we use as a proxy for tire wear. Tire wear, as I mentioned, is this complex mixture of a lot of different things. So people smarter than I am and more chemically oriented, have determined that the styrene butadiene rubber creates pyrolysates in the PyGCMS that we can quantify and use to sort of calculate what that means as a representative for tire wear.
Great. Is there any other question? Maybe if you also want to say a comment, something, if you want to raise your hands and speak, feel free to do so. I mean, we are almost at the end of the webinar. Really good to also see a couple of people have answered that they do some work related to microplastics. So yeah, somebody does work in sources distribution and ecological impact of microplastics. There is plastic eating microbes as a solution review. There is also someone using it as a potential measure for if water of health in water. Someone is working on microplastic pollution research at the Scottish Association of Marine Science. Someone works on plastic associated chemicals. Yeah, there's someone who wants to work in the field, not doing work on microplastics yet, but wants to work in the field. Someone works on the policy. I mean, that's a lot of people working in microplastics, and it's quite amazing to see. Yeah. And then there's a question to Robin. Is it possible that the tire wear might be made of the polysoprene or another polymer type?
Hi, yeah. So there are multiple standards that people use when trying to quantify tire wear. And when we use the styrene butadiene rubber, we have many sort of qualifying ions that we look for. The simple answer is, I don't know, it certainly could be, and we're just not picking up on that with our analytical methods, which is sort of a double-edged sword with any field-based microplastics research. You're always going to miss something. So we had to go with what was available to our lab with the resources available to us and the methods that work for our lab's processing.
Great. Is there any more questions, answers? Somebody, I think Robin, again to you, Robin, a good presentation. I have a small question. Why collecting 8 liters of water at each sampling site? Is it representative?
Another great question. We went with 8 liters for logistical reasons. So I did grab samples, meaning I just stuck my jar into the water and collected anything that came through, since using nets can exclude lower size fractions that I wanted to be able to include. However, that means that I had to collect and process a full, chunk of water. So 8 liters was the most that I could feasibly collect in a day, fit on a boat, take back and filter in the lab. Ideally, I would have like 20 liters. That'd be amazing. But you have to, again, have trade-offs with what is feasible for the time and money that goes into it. So 8 liters was just kind of the best that we could do. Thank you for that question.
Great. Yeah, there are no more questions, and there are no hands up. But I think this was a very great. I think okay. Now there's a question. Does the type of MP to microplastics like fiber, filament, film have different impacts? I'm sure this goes to both of you.
I can go ahead and start. So they do have different ways of being able to bypass different things. So when we think of, for example, wastewater, when we talked about that, the smaller fibers that are able to fit through the filtration systems are because they're fibrous, because they're able to get through those grates and other filtration mechanisms that they have in place. So in terms of fiber having that shape, it makes them more successful to bypass that filtration. And while they don't have different effects per se in terms of the shape specifically, we would be able to see more of the impacts on the type of plastics, so the chemical composition. While the shape can influence, in terms of the interactions with filter feeding animals, that is something that could be accounted for. But a lot of what we see in terms of the environment has to do with their chemical composition, and also things like their ability to serve as an adhered surface to metals, toxic chemicals, and of course, different types of bacterial colonies.
Yeah, I'll just piggyback. Lonnie did a great job of answering that. When we're talking about sort of fate in the environment, then shape can play a role, as fibers tend to be more buoyant with their long aspect ratio, whereas fragments tend to sink more rapidly. And as Lonnie said, the shape is going to influence which organisms are interacting with them. So, fish that are used to eating sort of spherical fragments are gonna maybe get more exposure to those than like a fiber or a film. But like Lonnie said, it's really the chemical leachates and that interaction that we expect to have the greatest impact when we talk about sort of ecosystem health.
Great. I think there's a long one here. Greetings. So thank you for the excellent presentation. My doctoral research focused on the intersection of culture and single-use plastic management in Yaoundé, Cameroon. I'm currently planning a study on the potential impact of degraded plastics and microplastics in underground water sources, with particular attention to the associated health risks for communities relying on such water for domestic use. I look forward to the possibility. Okay. This is not a question, but possible collaboration. Yes, so please, uh, from Valentine, right? Feel free to follow uh, Lonnie and Robin, connect with them. I mean, if you also have very specific, maybe questions or ideas that you would like to exchange, I can maybe ask them and give you their emails or ways to reach them so that you can connect better. Yeah, but all right. I think that is it. We don't have any more questions. Oh, I think there might be or no. Okay, yes, we don't. So great. Uh, Thank you, both presenters. Uh, People enjoyed the presentations very much. And I mean, the questions and the interactions show that they've really listened to you and they have been following, which is really great to see. So thank you again. Thanks to everyone who joined. Again, this is the ECOP program monthly webinars, and feel free to reach out to us if you have some very interesting topic around the ocean or the environmental space, anything related to the ocean, or and you would like to share, you would like to present on, whether your thesis, whether your work, uh, feel free to reach out to us, write to us, and then we'll be very happy to give you the opportunity to host you and give the opportunity to other ECOPs across the world to learn from your work. And again, we have ECOPs from 168 countries within the ECOP program, and 60 national and regional nodes currently, but also we don't have in all the countries, right? I was one of the co-founders of the ECOP Netherlands node because it didn't used to exist, right? And so if you always have the opportunity to establish or help establish one in your country if you do not have a national node yet. Just reach out and would be able to guide you and help you, support you to establish one, and then you can be a national coordinator. It's such a great opportunity. You can do a lot with it. You can check ECOP Canada, ECOP Belgium, ECOP UK. There are a lot of things they do with it. They give scholarships. They do a lot of capacity building programs. I think some governments have adopted the ECOP program. They fund it. There's a lot of amazing stuff that happened. There was recently the ECOP hackathon in Brazil, and it was very fun to see a lot of ECOPs, about 30 ECOPs funded there, and the hackathon. All these amazing opportunities are there for you to explore. So follow the ECOP program, but also engage in your national or regional node and play a key role. Feel free to always reach out again. Thank you for today. We have all very much taken some valuable lessons and knowledge from microplastics transportation from wastewater treatment plants and where they go to. And so thanks again, Lonnie and Robin. If you have anything to say, then maybe you would want to say it. But from my side, thank you and well done.
Yep, just thank you. Thanks for having us and thanks for attending.
Likewise. Thank you all.
All right. Have a nice rest of the day, evening, night, and see you another time. Bye.